Hello everyone, thank you for joining me today for this talk about the science behind these two products, Kefir-Nova and Kefir-Forte. Before I start, I'd like to acknowledge the priceless contribution and help from my colleagues at Silver Spring in Switzerland, as well as the team at CTC Life Sciences in the Republic of Korea. For those of you who do not know me, please allow me to introduce myself. I am 66 years old. I obtained my MD in 1980. I have a PhD in molecular biology and a specialization in diagnostic radiology. As a young postdoctor, I worked at Burroughs Wellcome in the USA, North Carolina, in 1984, and as a visiting scientist at the National Cancer Institute of the National Institute of Health of the US government in Bethesda, Maryland, USA, since 1987. Later, I was appointed professor of molecular biology at the University of Florence, Italy, or Firenze if you prefer, in 1992, where I worked until my retirement in 2014. I am the founder of a Swiss company called Silver Spring that is specialized in research and development of innovative products such as those that will be described today. One of my articles was sponsored by a Nobel laureate, Sir John Vane. My current research interests are in particular natural immunotherapy, quantum biology, and microbiome medicine. That will be the topic of today's talk. The science behind the Kefir-Nova and Kefir-Forte dates back to 2011, more than 10 years ago, when I began researching the effects of microbiome-based approaches on the immune system. Here on the left, you see the first article published in a peer-reviewed journal, the Italian Journal of Anatomy and Embryology, in 2011, where we describe the effects of this microbiome-based approach on the immune system, both in healthy individuals as well as in individuals with HIV, that, as you know, it causes immune deficiency. Now, these two products, for administrative and legal purposes, they're classified as probiotics. However, they are much, much more than simple probiotics. They represent, they support, they reconstitute an entire microbiome. So please, give me just a few more minutes to explain what an entire microbiome is and why it is different from simple probiotics. What are probiotics? Probiotics simply are friendly microbes, friendly bacteria, sometimes friendly yeasts, and these are friendly microbes that support our health. So in a microbiome, that actually is a reasonably well-defined habitat which has a distinct biophysiochemical properties as described in Wikipedia. So bacteria or eukaryotes like yeast, these are probiotics. So when you go and buy a common probiotic, what you buy are bacteria and eukaryotes. A microbiome is much, much more, because a microbiome is an entire ecosystem where, of course, you have the microbes, the bacteria and the eukaryotes, but also you have their theater of activity. That is all the molecules, all the information that they do produce for their own sake, but also to support the health of the host, that in this case it's us human. So in their theater of activity, you have proteins, peptides, lipids, polysaccharides, but also you have nucleic acids like DNA, RNA, mobile genetic elements like plasmids, including viruses, the viruses of the bacteria, the friendly viruses that are called bacteriophages or phages. And also you have internal and external structural elements like environmental conditions that are part of the microbiome. And you have microbial metabolites. Microbial metabolites are glues that are produced by the probiotics, again, for their own sake, but they also support our health. So talking about microbial metabolites, so these metabolites that are produced during the proprietary fermentation process of our products, we have discovered that in a typical kefir, there are 236 unique peptides that are formed during the fermentation of milk, and in our case, milk and colostrum, or as far as the kefir forte is concerned, during the fermentation of hemp seed proteins. So this article was published a few years ago in 2015, and it describes how in a typical kefir you find 236 novel peptides. Novel means that they were not present in milk, or in our case in colostrum or in hemp seed proteins, but they are formed by the bacteria as part of their microbiome during the process of fermentation. Process of fermentation that is a proprietary process, very innovative, that we have developed in our Swiss laboratories. Now, what do these 236 peptides do? Well, again, going back to a typical bovine kefir, like the one described in this article, they have a number of health supporting properties. They inhibit those enzymes that are responsible for hypertension. They are anti-thrombotic, so they help with blood circulation. Mineral binding peptides that help with the absorption of a number of elements, or also antioxidant peptides, antimicrobial peptides, opioid peptides, but most important for our purposes are the immunomodulating peptides. So those molecules produced by the bacteria in the context of the entire microbiome that help supporting our immune system, empowering our immune system. Now, we concentrated our attention on one particular protein produced by the microbes in our products. That is called the GC-MAF. GC-MAF stands for GC protein-derived macrophage activating factor. You can read a lot about this wonderful molecule, this wonder molecule. You can read in this journal that is called What Doctors Don't Tell You, where it is reported in a very easy way. And here the author describes this like a super molecule that is a key to healing a vast array of illnesses from autism to cancer. And if you want to go to the hard science, then you can go to the National Library of Medicine of the US government, the search engine that is called PubMed, and just look for GC-MAF, and you will find articles in peer-reviewed journals, highly reputed peer-reviewed journals, describing the promising role for GC-MAF in cancer immunotherapy, from the bench to the bedside, so from research to application, or the potential role of GC-MAF in COVID-19, or the role of GC-MAF in patients with multiple sclerosis. And if you like, you can go to PubMed, look for my last name, Ruggiero, and the GC-MAF, and you will find a number of scientific articles co-authored by me where we describe the effects of GC-MAF in a variety of conditions, from human breast cancer cells to human neurons and glial cells on angiogenesis and so on. So you can go and read all my past research on GC-MAF. But what it is important is that most of the articles that have been published on GC-MAF, they have used kind of a purified chemical GC-MAF, and they have observed all those excellent results. Sometimes they are described as wonderful results. In our products, we have 100 times more GC-MAF activity in comparison with that purified chemical GC-MAF. So our products, they contain 100 times more GC-MAF activity in comparison with the GC-MAF that has been described in most of the scientific articles that I have quoted before. But as I said, our products are much more than one single thing, are much more than a couple of probiotics, are much more than GC-MAF in itself. For example, they contain phages. And phages are the new frontier of therapy. In our microbiome-based products, we have hundreds of different microbial strains. And again, this is a big difference in comparison to the common probiotics you find in the market, where you have just a few strains. We have 100, between 300 and 400 microbial strains. And in these a vast array of biodiversity that is absolutely unique, we have several phages that have interesting health supporting properties. Here I just highlighted 3 sentences coming from this article. For example, phages are known to induce tumor destruction through macrophage activation. So again, here we have a combination of factors. The GCMAF on one side stimulates macrophages, and we have already seen its wonderful effects. But also in our products, as I said, we do not have only GCMF, we have a number of things, including phages that do the same. They stimulate macrophages, and it is known that phages, through stimulation of macrophages, they induce tumor destruction. And also one protein coded for by one of those phages that is present in our microbiome-based product, it is called SAC. It has a human equivalent that is called RAD52. And this has been demonstrated to suppress HIV-1 infection in a manner independent of its ability to repair DNA double-strand breaks through homologous recombination. So this phage, it is a lactobacillus phage. You can read all the details in that article. Not only codes for a protein that is involved in repair of DNA, which in itself is very important for repairing the damage exerted on a DNA by a variety of factors, from chemicals to radiations and so on. But in addition to this, and independent of this protein also inhibits HIV infection. Or, for example, another phage that is called Streptococcus phage TP778L may be of interest in the context of the phenomenon that is designated viral superinfection exclusion. So in other words, this phage prevents viral superinfection. Maybe this is the reason why those authors I mentioned, they quote phages as potential tools in the fight against other viral diseases, such as COVID-19, for example. And our Swiss-made microbiome-based products are the only ones reproducing the complexity of the human microbiome. So I think that you may begin to grasp the difference between our products and common probiotics. The difference is a difference in biodiversity, complexity, number of molecules, the number of agents that support our health in the most natural way. To conclude, Kefir-Nova and Kefir-Forte are based on more than 10 years of research in the field of microbiome medicine, and they are backed by a number of peer-reviewed articles. And this can't be said for the majority of probiotics. But they are much, much more than probiotics, as I say. They are manufactured in a state-of-the-art facility in Switzerland using proprietary innovative methods of fermentation. And as I said more than once, they are much more than simple probiotics. Rather, they are entire microbiomes containing, among many other components, immune-modulating molecules such as GCMF as an example, and phages such as those that I've described before. In this presentation, I've highlighted only a few examples of the complexity and the potential applications of these products. I mean, I could talk for hours and hours describing each one of those 236 molecules, each one of those phages, each one of those plasmids that we have found being present in our extremely biodiverse and complex products. Overall, however, what's important is that this complexity helps supporting and reconstituting the human microbiome. And because of this, they may prove useful as a complementary approach in a number of conditions where support of immune defenses is needed. I thank you so much for your time and attention, and I hope to see you in the future with further information about these amazing products. Thank you.